All right, let's look at a controlled experiment. So let's define a couple terms. The first one is a control group. So the control group, this is a group in the experiment that does not receive the variable being tested. What's the purpose of it? So the results of the experimental group can be compared to this group to determine the effects of the variable being tested. Our second term, experimental group. This is the group receiving the variable being tested. What's the purpose? It allows the effects of the variable to be observed. So let's use an example. So let's think of plants. So you want to grow some plants. So I have here, this is a lady slipper that I had taken a picture of a number of years ago. And if you had some lady slippers or some other plants and you want them to grow well and you want to use a fertilizer. So let's say you want to figure out if the fertilizer is effective or not. How would you actually test that? So what you could do is you could set up a controlled experiment. So what I did here down below here is I drew two groups. On the left hand side over here, this is the experimental group. This one's going to receive the fertilizer that I think I want to use. So this is going to receive the fertilizer. And then I have a control group. And this control group is not going to have fertilizer. So there's no fertilizer. And why would I do that? Well, whatever results I have in the experimental group, I want to compare that to the control group because any effect that I see or change I see between the two groups should be as a result of the fertilizer. So if the plants grow bigger and they grow faster in the fertilizer group or the experimental group, then I know that's the effect of the fertilizer. But on the other hand, if there's no difference, let's say the plants look exactly the same in my experimental group and my control group, that means the fertilizer has little or no effect, or no effect that's measurable. In a successful controlled experiment, you want to make sure that only one variable is being changed, and the variable being changed in this case would be just the addition of fertilizer. So, for instance, all the other variables like light, the amount of water that I give both groups is going to be the same. The amount of light that both groups are going to receive are the same. The types of plants that I use are going to be the same. The soil is going to be the same. The pots that I use are going to be the same. So everything's going to be the same. The only thing that's going to be different again is that I'm going to give fertilizer to the experimental group, and I'm not going to give any fertilizer to the control group. All right? All right, thank you very much.